Hi, I'm Gurian Marks. I'm a senior at Bronx Science, an avid ice hockey player, a jazz sax and rock guitar player, and a budding neuroscience researcher. Over the past couple months, I've developed a project in the general field of connectomics and neuroscience. Now, the Connectome project is a worldwide movement in which scientists from all different countries and all different disciplines have come together to try to create a wiring diagram of the brain, or a map of the brain that shows how each neuron connects to each other neuron, how each neuron functionally identifies in a circuit with each other neuron in the brain. Now, this project is incredibly important because most neurodegenerative diseases, such as, say, Alzheimer's or other personality disorders such as bipolar disorder and schizophrenia, or other neurological things such as memory, are all inherently circuit-related and circuit-based. So once we begin to understand this connectome, we can begin to find a cure to all of these terrible diseases. However, in finding the connectome, there are several limitations. And the first is that all of the data that we have to process must be processed by hand. Now, the type of data that we use in order to find a connectome is electron microscopy data, in which you take the specimen. In my lab, we use the zebrafish, and slice its brain into incredibly thin slices, about 30 or 40 nanometers thick, and shoot an electron beam through those slices to create a very detailed picture of the brain. And then using all those very thin slices, you put them one on top of each other to create a stack of slices, or a stack of images that you can then trace through and interpolate to create a reconstructed neuron. But all of this has to be done by hand. You may be wondering why. Don't we have computer programs to do that? Well, kind of, but not efficient ones. The problem is that machine learning algorithms don't have the pattern recognizing ability to be able to differentiate different types of neural constructs close to the soma. So, for example, in connectomics, we want to find the axon because that's the only branch of a neuron that sends out a signal and may show us how one neuron may connect to a different, or one neuron or one part of the brain may connect to a different part of the brain and start to show us neural circuitry. The other branches are dendrites, which all collect signals and collect impulses from other neurons. So the problem is that these two constructs, axons and dendrites, look very similar to each other close to the soma, which means that these machine learning algorithms can't differentiate between the two different types of branches and can't effectively make a connectome. Now, this is where my project comes in. So what I did is I made a plasmid which has a series of genes on it which encode a series of proteins all fused together, hence the name of my title. It's a fusion protein which localizes itself to the axon initial segment. It localizes itself because of a protein that I chose called ankyrin G, which stays only in the axon initial segment, which means that I'm only putting tags on the axon and not the dendrites. So what I'm doing is making sure that I'm making sure that these machine learning algorithms can now use that tag as a guidepost to differentiate between axons and dendrites and more effectively reconstruct neural circuitry. So in doing this, it's expediting the process of creating a connectome. And I'm incredibly interested in this because I've experienced firsthand all of the different effects of these neurodegenerative diseases that the Connectome project is trying to alleviate. I've experienced firsthand a loved one's major memory loss, and I can fully understand the greater emotional and social and economic distresses that come from these diseases or disorders in neural circuitry and memory, because I know these ramifications, but I'm excited to expedite the process of finding a connectome to be able to mitigate these horrible diseases and find a cure to them. That's one of the reasons why I chose that protein that I mentioned earlier, ankyrin G, because I found a paper that cited that in many abnormal animals, it was found that ankyrin G migrates out of the axon initial segment and accumulates into beta amyloid plaque, which is the major cause of Alzheimer's. So what I found is that I can use this protein in two different ways. The first is I can expedite the finding of a connectome, but I can also use this protein to possibly lead to ankyrin G-mediated therapeutics by being able to trace the production of beta amyloid plaque and thus Alzheimer's with my new construct. So I'm very excited to be able to be a part of this worldwide and interdisciplinary project, and I hope that you consider my application seriously. Thank you.